Welcome everybody, and thanks for joining us today to learn more about our next generation CAR-exosome therapy to target and treat solid tumours. We will have a Q&A session at the end of the presentation. So slide 3, please, Greg. INOVIQ is a biotechnology company pioneering next generation exosome diagnostics and therapeutics to improve patient outcomes in cancer. We have a patented exosome technology platform that has applications across research, diagnostics, and therapeutics. Our exosome isolation tools are commercially available via our global distribution partner, Promega Corporation, based in the United States, and is generating initial revenues. Our product pipeline includes 2 clinical stage diagnostics for ovarian cancer screening and breast cancer monitoring, and we have a preclinical stage exosome therapy in development for treatment of triple-negative breast cancer. We recently appointed a new Chief Commercialization Officer to sharpen our focus on partnering and strategic acquisitions as we advance our pipeline towards key development and commercial milestones. Our leadership team has expertise across exosome science, diagnostics, drug development and commercialization, and we're backed by a world-class advisory board to execute on our strategy. I'll now hand over to our Chief Scientific Officer, Professor Greg Rice, to provide an overview of our CAR-exosome program, how they work, their potential advantages over cell therapy, our supporting data, and also our development plans. Over to you, Greg.
Thanks, Leanne, and again, welcome everyone to the webinar. What I'm going to do is to focus on INOVIQ's R&D priorities and our lead products in development, of which there are 2. Firstly, the ovarian cancer screening test. Now, while this is not the main topic of this webinar, I do want to make a few high-level comments just to inform our investors and the market. INOVIQ, over the past 4 years, has been working with the University of Queensland to develop this much-needed test. There's a clear clinical gap. There is no approved screening test for ovarian cancer. So most of the ovarian cancers are diagnosed at a late stage when the survival rates are poor. The challenge, the challenge, as with any clinically relevant screening test, be it ovarian cancer, breast cancer, prostate cancer, has been to develop a test with a high enough specificity to be useful. The specificity of the test tells you how good the test is at correctly identifying women who do not have ovarian cancer. The higher the specificity, the better the screening test. We don't want to be telling women that they have cancer when they do not. For ovarian cancer, the specificity required by clinicians is a minimum of 99.6%. And this is to minimize those false positive rates and the unnecessary surgical follow-up resulting from a positive diagnosis. INOVIQ will be reporting progress on the development of its ovarian cancer screening test at the annual meeting of the American Society for Clinical Oncology in June. So look out for that update and we can then inform the market of our progress there. The second lead product that we're working on is currently a targeted treatment for triple-negative breast cancer. And this is really the first therapeutic product that INOVIQ has been developing. Over the past 2 years, INOVIQ has been building a platform technology to deliver next-generation targeted therapies. We now have the right people. We have proprietary technology to develop targeted cell-free or acellular therapies. So what does that mean in lay terms? I think one of the most significant recent advances in treating cancer has been the development of cell immunotherapy. And this is where white cells are collected from the blood. They're modified in the laboratory to hunt down and kill cancer cells. Those cells are then grown up sufficiently to generate a treatment dose, and then they are injected back into the patient to seek out and kill cancer cells. And this is called, for example, CAR-T cell therapy. This approach works well for blood-based cancers, but not so good for solid tumors. And Professor Miles Prince has been one of the pioneers and leads in clinical trials of this type of approach for treating blood cancers in Australia. And Professor Prince is a member of our Medical and Scientific Advisory Committee. Professor Phil Darcy, who is also a member of our Medical Scientific Advisory Committee, is one of Australia's leading clinical researchers in CAR cell therapies, and we'll hear from Phil at later in this presentation. What INOVIQ is doing is really taking it to the next step to try and overcome some of the problems associated with CAR cell-based therapy. And what we're doing is we are using a white cell, an immune cell called a natural killer cell. These do not come from the patient. We actually immortalize them so that we can grow these cells up and generate a cell bank so that we can actually use these cells for everyone. We're able to reprogram them. So the same as in cell therapy, we reprogram to target and seek out cancer cells. In this case, triple negative breast cancer cells. We grow them up exactly the same way as for cell therapy. But rather than injecting these cells back into the patient, we isolate and collect the cancer-killing exosomes that are released by these cells. We then are able to generate this CAR-EV-NK therapy and inject them back into the patient. Now, this approach has many advantages. As you can see here, for example, we're using this approach, we are able to treat many patients rather than a single patient. So it's we're able to supply multiple doses to patients and also use combination therapy. So these are some of the advantages of this type of approach. It's an allogenic approach, so we're not using patient samples, we're using an immortalized cell line to generate this product. So why focus on triple negative breast cancer? Again, it's because of clinical need. There is no approved targeted cell-based therapy for triple negative breast cancer. And that's because it lacks the three most common drug targets for treating breast cancer. That's the estrogen receptor, the progesterone receptor, and the HER2 protein. This means that there is limited options in terms of trying to treat triple negative breast cancer. Hormone therapy and treatments that target the HER2 protein are ineffective. And chemotherapy remains the most common treatment for triple negative breast cancer. Initially, most of those triple negatives respond well, but often resistance to chemotherapy generates, leading to relapse. So there is indeed a clear clinical need for a targeted therapy as an alternative approach to chemotherapy. The target that INOVIQ is focusing on is the EGFR protein that's expressed on the surface of triple negative breast cancer cells. EGFR is expressed in most triple negative cancer cases and much higher than we see in other types of breast cancer. And our therapeutic agent is the cancer-killing exosomes that target EGFR expressing triple-negative breast cancer cells. And remember, the way that we achieve this targeting is to add to the surface of those exosomes a protein that binds the EGFR that is expressed by the cancer cells. Once the exosomes bind to those cancer cells, they release bioactive molecules that lead to cancer cell death. It's probably useful to try to understand a little bit more about the exosome itself. Exosomes are tiny packages that are released by all cells, and they're involved in cell-to-cell communication. They're about 200 times smaller than a normal cell, and they're in extremely high concentrations in blood, trillions of them per mil of blood. If you like, they're like microscopic delivery trucks. And if we cut one in half, we can see that they actually contain bioactive substances such as proteins, RNA, fragments of DNA, and other molecules. The exosome is actually protected by a membrane. This allows it to move safely between cells and protects the contents of the exosome. INOVIQ's exosomes carry cancer-killing proteins, and they are delivered to triple-negative breast cancer cells by the expression of the EGFR binding proteins that we include on the surface of those cells. So this allows us to deliver cytotoxic cell-killing agents to specifically cells that are overexpressing this particular type of EGFR that triple-negative cancer cells express. Just in a little bit more detail, this is a process that we're currently using in the lab. We are modifying both T cells and NK cells. We're putting on the surface of these cells proteins that bind to the EGFR on cancer cells. Those binding proteins are also inherited by the exosomes, or EVs, extracellular vesicles, that are produced and released by these cells. Those exosomes contain cytotoxic agents, killing agents such as perforin, granzyme, cytokines, other proteins. These exosomes then are able to target cancer cells that express the EGFR on their surface. They release their cytotoxic cargo and induce apoptosis or cell death in the cancer cell. The other advantage of using these small packages, these exosomes, is that we are able to load them with other chemotherapy agents, with peptides and RNAs that suppress the activity or kill cancer cells, and we're able to use them in combination therapies with, for example, even cell therapy or antibody treatments, or indeed even cytotoxic agents. So what have we actually done in the lab? We've been completing some proof of concept studies around our immortalized NK cells. So these are not patient-derived cells. They're ones that we have in a bank that we can pull out and grow up any time. The target is the EGFR. We're able to use our proprietary technology, our XOS technology, to isolate therapeutic grade exosomes for analysis. We obviously run the quality control and characterize those exosomes to ensure reproducibility of the product. And then we're able to evaluate the killing activity of these exosomes in the lab. And what we've been able to do is to show that we successfully incorporate this EGFR targeting protein on the surface of those NK cells. The target is fluorescence, so that we can actually monitor its incorporation into NK cells. We've then been able to run it through our proprietary isolation technology, demonstrating that we also see those cytotoxic compounds present in the exosomes. This material is then used for therapeutic use. What we've been able to show in the laboratory is that these EGFR CAR-EVs kill breast cancer cells. So shown here, the lower the number, the less number of cells surviving. So here we see we're able to, in untreated versus the EV treated, we're seeing a decline in the number of cells. We've also been able to show that it's effective against blood cancers, less effective because they're expressing less EGFR on their surface. More recently, we've been able to demonstrate that these extracellular vesicles are also effective against triple-negative breast cancer cells in the laboratory. So here we see a dose response, increasing number of exosomes, EV killing activity, and the number of cells declines with treatment. So that's the proof of concept studies that we've done in the laboratory. What we're now planning to do is to start the program with Peter Mack and Professor Phil Darcy to again evaluate the anti-tumor effect of these CAR-EVs on triple-negative breast cancer. So Professor Darcy will take the same approach. This will be done by Peter Mack, so it's independent of the work that we have done in the laboratory to modify these CAR-NK cells and to isolate them. The isolation will actually be done in our laboratory here using our proprietary technology. Peter Mack will then generate cells in vitro in the lab, and we'll be able to test the killing activity of these exosomes on triple-negative breast cancer cells. On successful completion of that study, we will then again move to complete an in vivo study using a mouse model. Again, with the Peter Mack, we will perform an experiment to evaluate the dose of extracellular vesicles required to reduce or kill the cancer and how long it takes. And again, this work will be done by Professor Darcy's group. So this is really a summary of the path to market, going through this process of engineering the CAR cells, isolating the extracellular vesicles, confirming their in vitro and in vivo efficacy in killing cancers, leading to an investigational new drug application and clinical trial. Some of the major benefits of CAR-EV therapy over cell-based therapies includes their improved safety profile. They are less immunogenic than cells themselves, and they don't obviously result in secondary tumors. They have no capacity to multiply. They have improved efficacy. They're able to penetrate places where cells can't. They can cross, for example, the blood-brain barrier, which opens up the opportunity to develop therapies down the track for example, brain tumors. We're able to give multiple doses to a single patient on their own or in combination with other therapies. This is again an advantage over the current cell-based therapy approach. We're able to deliver them to multiple patients, so the same preparation can be delivered to multiple patients. Again, an advantage over cell-based therapy where a single dose is for a single patient. So this means that they are more cost-effective, they're cheaper, they're faster, and they have advantages in terms of obviously the supply chain. And ultimately, we hope lower the treatment costs and improve benefits to patients in the healthcare system. So just to summarize before I hand back to Lian, this is what we've been doing in terms of the EV diagnostic program, therapeutic program. We've been able to generate our modified immune cells, develop the master cell bank. We have proprietary technology to isolate these extracellular vesicles. We've demonstrated proof of concept data, both with T cells and NK cells, showing that they're killing triple-negative breast cancer cells. Now, this approach is not novel in the sense that other people are already using this approach, using, for example, mesenchymal stem cells, T cells, and NK cells. In fact, MD Anderson has taken this type of approach through to phase 1, 2A clinical trials. So it's a proven technology and de-risked. We have this operating in our laboratory, and Peter Mack is well versed and experienced in this process. And this is really the next step that we will take in collaboration with Peter Mack, developing the preclinical package to allow us to go to IND submission. And I think there I will hand back to Leanne.
Wonderful. If you can move to the next slide. Thank you very much, Greg, for that excellent presentation. I'm very pleased to introduce Professor Phil Darcy. He's the co-leader of the Cancer Immunology Program, group leader of the Cancer Immunotherapy Laboratory and at the Peter MacCallum Cancer Centre, and NHMRC Principal Research Fellow. Professor Darcy's focused on novel T-cell-based immunotherapy approaches for treating cancer in both preclinical mouse models and clinical translation. He's a member of INOVIQ's advisory board, as Greg has already mentioned, and will be leading our CAR-exosome collaboration with Peter Mack. I'll now hand over to Phil, who will tell us about his background and key research interests.
Thank you, Leanne, for that introduction. As Leanne mentioned, I have a lot of experience and expertise in development of CAR T-cell-based immunotherapy, and in fact, over 30 years experience. And just a couple of highlights. We were the first team to run a CAR T-cell trial in Australia back in 2010. And so we've had over 30 years experience in development of this type of therapy. Other notable highlights is that we've been able to disseminate our research widely in prestigious scientific journals such as Nature, so it's testifying to the significance of our work. We're very excited to join INOVIQ and develop their CAR-exosome technology and apply our expertise in this area to test this in our preclinical models. The CAR-exosome technology, as just mentioned previously by Greg, is a next-gen acellular therapy. It has clear advantages of safety and efficacy over cell-based therapies.
Greg, could you move to the next slide?
Yep, sure.
And has a lot of potential for treatment of solid cancers. So this slide here just really lists the number of advantages of CAR-exosome therapy and overcoming many of the hurdles that we're confronted with using standardized CAR T-cell therapy so far. So it's a versatile and flexible technology platform, has many therapeutic applications. One of the advantages is that these EVs show exquisite specificity for the tumor target. They inherit the targeting specificity from the parent CAR-NK cells, as mentioned previously. One of the important advantages is that these EVs are derived from the CAR-NK cell, is that they lack PD-1 expression and can avoid suppression by tumors that express this PD-1 ligand called PD-L1. This PD-1, PD-L1 axis is one of the major suppressive pathways used by tumors to avoid immune recognition. So that's one of the advantages of EVs. Then they don't become exhausted and they can continuously have anti-tumor activity, unlike cell-based therapies, which the T cells that are transferred into patients become exhausted very quickly. As also mentioned by Greg, these EVs have very potent anti-tumor efficacy. And they do this by delivering cytotoxic molecules such as granzymes and perforin, which are contained within these EVs. Another, I think, very huge potential of these EVs is we can load them with various chemotherapeutics such that are used to treat patients. And the idea here is that EVs can actually deliver these drugs specifically at the tumor site, and this shows they can target the tumor specifically without having these off-target effects. One of the clear advantages of this exosome technology is it's safer than standardized CAR T-cell therapies. There's a reduced risk of immune rejection cytokine release syndrome and graft versus host disease, which are commonly seen with CAR T-cell based therapies, and it presents a huge problem with this type of therapy, the treatment of patients. So these EV technology can overcome some of these major hurdles. They're also short-lived with transient activity, and again, this reduces the risk of this sustained immune activation or exhaustion, as I've mentioned. One of the key advantages of using this approach, I see, is that we can use it as an off-the-shelf therapy. We can't do this with standardized CAR T-cell therapy today. It's an autologous system where, a personalized treatment, if you like, where you can only treat one patient. This development of this technology has the potential to treat many patients as an off-the-shelf therapy. So we're very excited to work with INOVIQ to develop this technology and test the potential of this therapy in our preclinical models with a view of treatment of triple-negative patients in the future. Thank you.
Thank you very much, Phil. Next slide, please. I'm now very happy to introduce our new Chief Commercial Officer, Dr. Emma Ball, who actually starts with us next Monday, which we're very excited about. Emma joins us at a pivotal period as we're actually advancing our ovarian cancer screening tests towards clinical trials and our CAR-exosome therapy into in vivo, so animal efficacy studies for breast cancer. Her expertise in business development, commercial strategy, and building pharma partnerships will be instrumental in driving the strategic partnering of our sub-B2M and exonet technologies, expanding our exosome business, and delivering our diagnostics and therapeutics to patients. I'll now ask Emma to tell us about her background.
Thanks, Leanne. Thanks for the very warm welcome to the team at INOVIQ. So a bit of background, I'm a biotech commercialization expert with about 25 years of experience in strategic and operational roles spanning across therapeutics, vaccines, and diagnostics. Most recently, I was Global Head of Ecosystem Development at Illumina, which is a US-based genomics and multi-omics leader. And I was responsible there for strategic partnerships, including with academia, governments, and other biotech companies to really embed genomics into the diagnostic and precision medicine ecosystem. Before I was at Illumina, I was at CSL for 15 years in various leadership positions in business development, licensing, corporate strategy, commercial development, and R&D project management spanning those 15 years. In my more recent roles at CSL, I led search and evaluation, due diligence, and negotiations for multiple transactions, including research collaborations, co-development, in and out licensing deals, mergers, acquisitions, and divestments, and across a number of different disease areas and technologies.
Thanks, Emma. So tell us what excites you most about joining INOVIQ and what will be your key focus to drive our business forward.
Yeah, so look, I'm thrilled to be joining at such a pivotal time in the company's growth. I mean, I think we've heard a really exciting development today. And I was really drawn to INOVIQ's diverse portfolio of diagnostics and therapeutics and research tools underpinned by this novel exosome technology platform. So I love the optionality of the portfolio, and I'm really excited to leverage my commercialization and partnering expertise to accelerate development and accessibility of these innovative diagnostics and therapeutics for cancer and also other critical diseases. So initially, my focus will be on the strategic partnering of the sub-B2M and exonet technologies and commercialization of our ovarian cancer screening test and expanding our exosome business. So I'm really looking forward to engaging with INOVIQ's investor community in more detail once I officially commence in the role shortly.
As are we. So we're very much looking forward to you starting on Monday. And thanks for joining us today. Next slide, please. So this slide just gives us a quick overview on the exosome therapeutics market. There are currently no exosome therapeutics that are registered and commercially available on the market. The first exosome therapeutics are expected to reach the market in 2029, growing the market from around 32 million in 2029 to 1.4 billion in 2030. As INOVIQ is developing CAR-exosome therapy for treatment of solid tumours, we're really part of the CAR T cell therapy market. I think they'll end up calling it the CAR cell therapy market, which is expected to grow from $4.3 billion to $61 billion by 2034. In terms of the exosome therapeutics market, it is rapidly expanding. There's been over $570 million in investment over recent years. There's 120 EV therapies in development across a range of indications from oncology, regenerative medicine, autoimmune disease, and neurology. The table on the right actually shows you that there's 10 exosome therapies that are currently in human clinical trials. And that means that safety and efficacy has actually been initially proven for a number of different cell types that have been used to produce these exosome therapies. There's also a plethora of preclinical studies that are underway, including for NK cell-derived exosomes, which is what INOVIQ is working on, that has shown initial preclinical safety and efficacy in animal models. Given that we're working in the breast cancer market, you can see there's 2.3 million new cases of breast cancer every year around the world. That includes 25 million women living with breast cancer. Our target is the 10 to 15% of those women that have triple-negative breast cancer where there are no targeted therapies. Next slide, please. This slide provides a summary of early-stage exosome and cell therapy deals. It highlights many of the transactions have large total deal values of around $1 billion and include upfront payments of 20 to 100 million, R&D costs, milestones, and double-digit royalties. That actually concludes our presentation for today. I hope you enjoyed it and are as excited as we are by the potential of a CAR-exosome therapy to target and kill solid tumours. I will now hand over to our CFO, Mark Edwards, to host the Q&A session.
Okay, thank you, Leanne. We haven't had any questions come through, but now serves as a perfect opportunity to shoot those through on the platform, and we'll respond to those as best we can. Otherwise, if you'd like to ask a more discreet question, we'd certainly invite them via email, and they can be sent to info@inoviq.com. So we'll just give it a little bit if there's any questions coming through online. Okay. Nothing seems to be coming through at this stage.
I think that probably wraps it up then, Mark.
Yeah, I think so.
Thank you, everybody, for listening, and we will continue to inform the market of updates on our portfolio as we progress our programs across clinical diagnostics for ovarian cancer screening, breast cancer monitoring, and our very exciting CAR-exosome program for treatment of triple-negative breast cancer.
Yeah, sorry, Leon, we have one question coming in late, which I think would be timely to address. There's been a question around any results from human trials for exosomes in the list in the presentation.
There has been data across a number of those different clinical trials. We would have to discuss those separately, but essentially there's data from Duchenne muscular dystrophy trials, from acute respiratory distress syndrome. Again, these studies have shown both safety and efficacy in human studies. Those two drugs candidates are actually in phase 3 clinical studies, so they've had to have shown safety and initial efficacy. There's also a drug in development for ALS in phase 3. There's another candidate that's been in development for diabetic foot ulcers that's in phase 3. There's other studies with a cancer immunotherapy that's been developed using dendritic EVs, and so that's another immune cell that's in a phase 2/3 clinical trial, again having demonstrated clinical safety and efficacy in humans. And there's also early stage studies in pancreatic cancer, rectal cancer, and neurodegenerative diseases and colorectal cancer, actually, that have demonstrated initial safety.
Okay, thank you, Leon. Another question has come in. It seems the sub-B2M and XNX technologies hold incredible promise. How confident are you that this promise will be realised as commercial products being sold, not just therapeutics?
We're extremely confident in our product pipeline. Our ovarian cancer screening program, which Greg mentioned at the beginning of this presentation, has already delivered outstanding results that we announced in December. Since then, the company has been working with an expert in computational science to develop a multivariate algorithm using a number of different biomarkers that has shown us extremely outstanding results. As Greg said, we will be delivering a poster at ASCO on this in May. Additionally, we expect to announce further results around our ovarian cancer screening test before the end of this quarter. We believe these results will be very exciting. We're very confident in the ability to move that program forward. We also announced very recently that we have a publication for our what we call our new CA15-3 test, which is an improved CA15-3 biomarker test for monitoring breast cancer. One of the key things that came out of that peer review of the publication was the exciting potential of combining our glycosylated biomarker, which means a sugar attached to the biomarker CA15-3, with other biomarkers to potentially develop that as an early detection test for breast cancer. So we believe that these programs are extremely exciting and will continue to move forward into commercialisation.
Thank you, Leanne. I can confirm there's no further questions.
Thank you. I think we'll close the session now, Mark.
Yeah, I think so. Thanks everyone for their attendance, and to our panelists as well. Thank you.